Hello, thank you so much for taking the time to join. We're going to get started in a couple of minutes. We're just going to wait until probably around 11:02 a.m. to give some time for other people to join. Hello, for those that have joined, we're probably going to get started around 11:02 a.m. But we truly wanted to thank you for taking the time to come today.
Okay, I think we can start. Welcome to the Synapse Session. Synapse Session is a project of Simply Neuroscience and specifically of the awareness team. And our mission is to raise awareness about mental health and different neurological disorders. So please keep an eye on the chat for the links that will be sent out throughout the meeting. And I'm going to send out the paper right now that we're going to talk about today.
Here's some expectations for today's Synapse session. We ask that you please be respectful to presenters and attendees throughout the meeting. We kindly ask that you keep your microphone muted. Any questions can be asked during the Q&A session or they can be typed in the chat. Please use the raise hand function if you would like to ask a question. We encourage participants to have their cameras on to foster engagement, but we understand and respect your privacy if you choose to keep it off.
I guess I'll pass the microphone to Vishaka to introduce our guest speaker for today.
Yeah, sure. Today we have Aniqa Saiyara, and little about her background. She earned her MD in dentistry from Bangladesh, which served as a hint for her in her healthcare career. She was ultimately drawn to Poland as a result of her robust interest in research, which developed during her period in the medical field. At Jagiellonian University, she pursued her master's degree and cultivated an interest in neuroscience. It was a thesis which focused on unpredictable chronic mild stress that sparked her passion for understanding the complexity of the brain. She began her PhD journey in 2024 at the Match Institute of Pharmacology in Polish Academy of Sciences, with an emphasis on neurodevelopmental disorders such as ADHD, autism spectrum, and addiction. In future, she wants to expand her knowledge, particularly investigating the mechanisms of addiction and pursuing potential cures. So I will hand over to Miss Aniqa herself.
Thank you, Vishaka. I hope everyone can listen to me. I would like to start with the introduction part uh, about addiction uh, because we all only talk about depression. So I believe that depression and addiction are both very interconnected with each other because for addiction is a chronic mental health disorder which is associated with dysfunction of our reward system. Means like our happy mood, sad, our cravings, those kind of system, and which is characterized by compulsive, repetitive behaviors and an uncontrolled intake of addictive substance. It can be drugs, alcohol, like injection, different formulas, and the bad part is that the addiction, like 50% of in the addiction case, it relapse, and which can be triggered by different kind of stimuli. So on the right side of the my presentation, you will see this the pattern how it started from the initial use, then you get abused, then you get addicted, and this abstinence phase, which is the when you want to quit the drug, but you have this withdrawal and craving persistent due to the lasting brain dysregulation. And then you have more craving, and even if you want to quit, you can't quit, and it's come back again and it relapse. Which is very common, and it's triggered by the stress, cues, environment, and your daily lifestyle. And I would like to move to the next slide. You will see the data, the World Health data about the drug abuse. It's quite recent. If you go to that link, you can read. It's published in 2023. And when you see the number of the people who are addicted, it's quite a high. The highest number is cannabinoid, then opioids, then it's like morphine, pethidine, and then amphetamine, like ketamine, then cocaine. It's also high in number in Europe, and then in the last is MDMA. And when you think about the gender, of course, you see on the right side that males are more prone to get addicted, and they're more into this addiction related behavior, but females are lower in number but not that less. When you see the amphetamine, it's quite similar, like ketamine is quite commonly abused drug in Europe, and you can say in USA also. So I will move to the next slide, which is my topic for today. I choose this paper. I think you all get the link before. It's about paternal cocaine seeking motivation, which defines offspring vulnerability. We're going to talk about how children can get affected because of your father's addictive behavior, and which part of the brain it's affected more. Here in this paper, they're focused on in ventral tegmental area, and they are searching for the gene which is mainly responsible. It's gamma 3. Moving to the next slide, I will talk about a bit about this background of this paper. What they want to focus, it's about drug addiction, which is chronic relapsing disorder characterized by compulsive drug seeking and use. And they did animal studies to prove their point. And they also say that while maternal drug exposure has been widely studied, means we all have more data or paper about mothers, but we don't focus on fathers, that their abuse also can affect our children. And they prove that recent evidence shows that paternal motivation to seek cocaine rather than passive exposure, increasing the addiction risk. And they use the animal model, rat, and they want to prove their theory by creating this model, self-administration model, that it can affect the voluntary drug-seeking behavior. And in the end, they prove that finding revealed that paternal cocaine-seeking behavior downregulate the gene, which is GABA A receptor subunit gene in this ventral tegmental area. I will explain a bit more in the next slide of mine. Okay. So this aim of the study is mainly focused on this brain area. Why it's important? Because it's located in the midbrain, and which is responsible for different kind of behavior, dopamine release, reward, motivation, addiction related behavior, and also other neurodevelopmental disorder like autism, ADHD. For to analyze this brain region is very important for us, and why they choose this gene is also a very important gene for those disorders. It's like imprinting gene, you can say. The full abbreviation of this gene is aminobutyric acid, and it's inhibitory receptor of our brain. And when it dysregulate, the brain becomes more excitable, and then you have this cellular imbalance in your brain. So moving to the next slide, which is our main tricky part, methods. I would like to pay attention to the audience. So I will explain a bit slowly here. So they choose this male rat model, and they divided it into three groups. One is saline, which is the control group, will not receive any kind of drug. The second is, you will see cocaine self-administration. They're receiving cocaine, 5 milligram per kilo. And the last is yoked. What does it yoke means? It means they will receive the drug, but they will not decide. They will just receive it passively. So they did this cocaine administration for 30 days. It's quite a long time to get addicted. And then later, because the focus of this paper is to see into the children if they are prone to addiction. So they mated with females and they created the offsprings. This one is our F1. There, so you have to focus when I say F1, that means it's offspring or children. So, and how they do this whole procedure. So in the below of the skin, you will see it's FR1. It means that fixed ratio. One means they will press. In the end, I will show you the picture of this machine. So if they press the lever one time, they will get one injection, and then day by day you get more addictive. Then they increase the infusion number also. So you have to press more. FR5 means like you have to press more. You have to be more aggressive to get this drug because you already getting addicted. So it was for 10 days, and there is a break in 1 day break, progressive ratio, and then again it's repetitive. So it's for total 30 days each. If you read the supplementary data of this paper, you will read that they did it for 2.5 hours each day without any break. So it's quite a long process to get addicted. So I will move to the next slide. Here you will see two graphs, and it's F1. You have to pay attention that it's offspring's results. So the offsprings are already born, and now they also go through the same process to see if they are addicted naturally or not. So when you see this division of these groups, you will see that the blue one, which is our cocaine group, has higher number of drug session. I mean, you see those stars, that means it's more significant. They're pressing the lever more, they're taking more drug. Naturally, they are prone to addiction without any training. So So they didn't need any training to get addicted. It's just inherited. And even when there is no drug, you will see that the blue line is higher, because even there is no drug, their motivation is stronger. So they're pressing it more to get the drug. So I will switch to the next slide. Okay. So here, uh, after this self administration process in the offspring, also they did this cocaine administration in the children also, just to keep this balance for 10 days. And then they sacrificed the animal and they took out the brain area, our focused area, and they tried to create a map, or you can say gene modules, which is bit tricky to perform, because you have to know which gene or which module you want to target. So they performed this sequencing, and they divided it into 4 colors. So this WGCNA means it's weighted gene co-expression network analysis. So it's a protocol they maintain to find the similar gene which is related to addiction. So they divided into 4 colors: the red and brown, which indicate your inherited gene, which is more stable kind of gene. You get those genes from parents. So this blue module reflects how brain respond after drug exposure, and the green module, it shows that how you have this specific motivation or not. And you if even if you don't understand all, just if you understand the red one, it's our main strong marker to understand the down regulation part of this gene. I will move to the next slide. uh, okay, so this part you will not find in the paper. I just calculated those gene, how many gene they analyze, and just to make you understand. And when you see that, because in offspring they divided into control group and cocaine group. So naive means control, they didn't receive any drug. So when you see the down regulation and this yellow marking, it's 285. It's not much. The genes are not reduced. But after cocaine, it gets higher. That means you are overexpression of genes. And when you see this dopamine, DEG means dopamine related gene, it also expressed much higher. So that means that you have problem with expression in the gene. So what does it mean? It means that it suppressed your neuroplasticity in the brain, and also it can alter the dopamine signaling also, and reduce the expression of synaptic and or metabolic gene. So overexpression, generally it's not good after any kind of substance. That means your inhibitory genes goes down and your excitatory genes or receptor goes higher. So you will act weird, or you can say in an easy way that you will be hyper, you will be impulsive. So I will move to the next slide. So the last experiment they did is this knockout experiment. It's also quite interesting. They want to prove their theory that this gamma 3 gene is actually, if it's downregulated, then it's actually influence your behavior. So what they do, they want to knock out or kick out, you can say easily, this gene by injecting one virus in offspring. So you can see in the below, there is a written virus infection. And they did in the 14 days of the offspring. They're very small. And then after few days, they did the surgery to put the catheter for the drugs. And then OFT means it's one kind of behavioral test they do, open field. And then they perform from this whole self-administration or drug-seeking behavior protocol for 10 days. So here they have two genes. It's like one is virus for this GABA3 and one is control. So if you see the graphs, you will see that even after knocking the gene, which in the red mark, you see that signalings are quite higher in FR5. That means that if you if this gene is not present, so you are motivated more. So they want to prove that this gene is really downregulated when you take more drugs in the drug session and also no drug session. But when they do this behavioral testing, open field or any kind of hyperactivity behavior, they didn't see any changes. So in the end, they see that they showed stronger motivation in the F1 rats, and lowering this gamma 3 actually increases your drug seeking behavior, which affects due to your addiction kind of behavior. So I will move to the key takeaways. I hope I can make you understand that offspring addiction risk is driven by paternal drug seeking motivation, not just drug exposure. I mean, it's not only depend on you, what you get from your parents, it also can lead to you this addiction like behavior. I mean, it can go generation by generation also. And they really try to prove that this gamma 3 gene in this ventral tegmental area actually increase your cocaine seeking behavior in your children. And addiction vulnerability can be inherited. It's not 100% proven yet, the paternal one they try to prove. And this, if you know which gene you want to target, I think you can manage to find a treatment also if you know which gene is responsible for addiction, because till now we don't have a treatment for this kind of strong drug abuse behavior. So we just have some supportive therapy, but still we don't have any drugs for in this relapse stage. So it's for us, it's very important to find out. It's just they're talking about one gene. I believe there will be more gene which is related if we understand the mechanism. So I think we can find out the treatment for future also. So I think I finished my paper part, and I want to show you what I do in my lab also, this self-administration machine or setup, what it looks like. We put the animal inside and close the door, and on the right side you will see those levers. I mean, it's funny that like rats are so smart, they can learn so easily. They easily press it with 2-3 days training. They learn how, if I press it I will receive this drug. And there is two light, so they remember with this light, whenever they press the light sparks, so they remember this part. And this channel is the infusion line for the drugs. And then in the next slide, I will show you some animal pictures to, I hope everyone will be okay. Okay, so this one is after surgery. Like we our main targeted area is right jugular vein because it's directly connected to your, it's very important vein for changes in your cardiovascular system and other peripheral system also. So we did the surgery, and in the middle of the picture you can see this is the cocaine syringe, because we cover it with foils because these substances are very sensitive to light. That's why people who are actually addicted, they are very sensitive to light and light and sound too. And this is our rat, how it looks like during this administration procedure. They're always connected, and from just the difference from the paper is that we do it for 6 hours for 21 days, but they did for 30 days. For us, 21 days, it's enough to make them addicted. Okay, on the next slide. Yeah, so I think the discussion part will someone will carry or uh, someone will carry the discussion or
I believe one of the awareness team volunteers might be having some connection issues.
okay,
But
okay,
I guess kind of just going over highlighting the main takeaways from the paper. So when looking
yeah,
at the paternal addiction or the paternal influence on addiction, so the studies confirm that fathers with drug addiction pass on a higher addiction risk to their offspring. So importantly, when fathers actively seek drugs, it has a stronger effect compared to passive exposure. And for the second point, with motivation versus other behaviors, despite the addiction vulnerability, offspring don't show differences in anxiety, learning, or movement, suggesting that changes are specific to motivation circuits. And with the role of epigenetics, the drug use and stress reshape Sherm's epigenetic marks like DNA methylation, which in turn affects brain gene expression in offspring, especially in reward related areas like the VTA. And with the transcriptome findings, sequencing the VTA identified gene groups involved in synaptic connections and brain signaling pathways. So this helped explain how addiction traits are passed down. And with the GABA3 as a key gene, so it was linked to psychiatric conditions like autism and alcohol dependence, as it was identified as a major player. Its reduced expression in offspring's brain neurons increased their motivation for cocaine. And with the validation of its role, when researchers lowered the GABA RG3 in control rats, they showed the same addictive behavior seen in offspring from addicted fathers, proving its crucial role. And for the final point with future directions, genes like MYT1L and other transcription factors that regulate membrane proteins and GABA receptors likely influence addiction susceptibility. So more research is needed to explore how these networks control drug-seeking behavior and dopamine release in the brain. Um, Miss Anika, if there's anything that you would like to add as well.
I think I would like to add that it would be better, like for future, if we see the comparison between mother and father, and then I think we can surely say that if actually paternal, which one has more, more influence on offspring.
So the final part that we have are some discussion questions. So I'm gonna pass the microphone to Tanvi to give us the first question.
So to provide further insight into the paper, oh, sorry. Okay, so the first discussion question is, what are some potential limits and biases of the research? You can feel free to either raise your hand or put it in the chat.
I believe Caroline has their hand raised.
Okay, Carolyn, would you like to unmute and answer the question?
Sure. So something I was actually curious about related to this question was the focus is really on male offspring because that did seem like a significant bias in the ability to generalize the results, especially given that there's already a gender gap in a lot of animal model research.
Mm-hmm.
So I was really curious about why that might have been the case as a decision by the research team, and how much that would potentially limit the ability to generalize how all these results might apply to women in general.
Mm-hmm. Um, yeah, that's a great question, and it's a really great answer too. Do we have anyone else? Okay, someone said, how do we know that this research isn't just specific to rats, but can also transfer to other animals or humans?
I
Okay,
think we can, I would like to answer. I think when it comes to rodents, like it's mice or rats, but you can do in other species also. But I think it depends on the country to country, because in Europe we can't do in monkeys, which is more similar to human, so it's more relevant. So I think we can do in other species too.
okay. And Ilya, would you like to answer the question?
Yeah, I think that from basis on what exactly I've heard about in the research is that it's mostly on gender models, like how exactly it could affect different genders at the genetic level, as well as the moods in psychology and the moods in, that affect the brain. Although I guess if I'm not mistaken, eventually, I guess.
Yeah, okay. These are all some really great answers. So some things to consider are the animal model limitations. So since the study uses rats, it's difficult to completely capture the complexity of human addiction, especially with the fact that rat behavior cannot truly model human social and psychological factors that affect addiction risk. And in addition, the study only focuses on paternal cocaine exposure and does not consider maternal contributions or the interaction between maternal and paternal factors. Similarly, the experiment only studied male offspring, which means that the findings may not translate to their female counterparts who might be affected by other factors, including hormones and structural differences. I think we can move on to the next discussion question.
So the next discussion question that we have is, if these findings were applied to humans, what do they suggest about intergenerational effects of drug use? So again, if anyone would like to raise their hand, come off on mute, or type their answer in the chat, we'd be please feel free to do so. Yes, Caroline.
Well, it suggests that potentially more so than exposure to drugs in kind of the parent generation, the extent to which an individual's parents actually sought out drugs and kind of engaged in those motivational behaviors, is more of a significant risk factor. So it could potentially change how that kind of risk is assessed, at least as far as like surveys and other kind of measures of assessing risk factors for potential offspring.
Yeah, that's a really great response. Ilya, do you have an answer?
Yeah, like the addition of how exactly drugs are being used and how do they exactly are affecting on the animal models itself, what physiological or again neuropsychological responses are being made, and exactly how exactly can this also be affected through the offspring itself. So other, I guess, maybe other more sophisticated approaches could be how exactly do these structures that exist in this drug affect the entire regions, and how exactly it could these regions at the genetic level could affect the motivation and the moodness of those animal models. Not just in the animals, but also human beings can do that as well if you consider all of these factors in the human models too, instead of just animals.
Yeah, that's a really great insight. So I definitely agree with many of the things that were stated. If these findings were to be applied to humans, they suggest that a father's drug-seeking behavior, even before the baby is conceived, could influence the brain development and susceptibility to addiction of his children. And another important aspect to consider is how the findings support the notion that the intergenerational effects of drug use are not just limited to genetics, but also may be impacted by epigenetic factors, especially those that are transmitted through changes in sperm that affect how specific genes work in offspring. And moving on to the final discussion question.
So the final question is, what further experiments would you propose to strengthen the findings? And if anyone has an answer, they could just unmute themselves or raise their hand.
I think one of the most obvious solutions could be also testing this on female rats to further the gap between the two.
Yeah, we like for example, some experiments that could be done could basically include testing female offspring and determining whether they also display similar changes in addiction-related behavior. Other experiments could include human correlational studies to basically see if similar changes exist in people that also have a history of cocaine usage. And also, we could also test other addictive drugs to determine if the findings can be generalized to other substances, for example, opioids. And lastly, you could also conduct an experiment that includes testing the reversibility of the effect by exploring if interventions such as behavioral therapy and GABAergic drugs can reduce susceptibility in offspring. And I noticed there was a question in the chat or an answer. This person said we could widen the research pool on different types of animals to see if this continues. I think that's a great answer.
I would like to also mention something else. If we could to just improve and strengthen the findings, we could do a more transcriptomic and multi-omic sequencing to not just only understand the effects on how exactly these drugs are affecting GABAergic and GABA neurotransmitters, but how exactly do these neurotransmitters are affecting, as you mentioned, somebody mentioned at the epigenetic level as well. How exactly then this will affect the epigenetics that will go to the mood swings, to these new processes, how exactly it can disrupt and degenerate at the genetic level if we're speaking the neurotransmitters and the neurons themselves, and how exactly can we find cure? Because after all of these drug addictions that all occur when they affect the neurotransmitters, not just at the neurological but genetic level, they need some cures. And since we don't know how exactly access which protein is which one is not correctly responding to the normal neurotransmitter states, so maybe we could focus more on the neurogenic level as that as well.
Yeah, I think that's very insightful. Caroline, if you'd like to add on.
It would also be very interesting to see if there's any evidence of the same kind of genetic manipulation in humans for behaviors that are considered less classic as addictions. So, for instance, like video game addiction, cell phone addiction, sex addiction, these addictions that are really tied to motivation seeking behaviors rather than to a specific substance, because the emphasis is so much on the behavior in this experimental model.
Yeah, that's a really great answer. I think it would definitely be interesting to explore other types of addiction, like game addiction or cell phone
Yeah.
addiction. So, thank you so much. Moving on. So, I know some people had some questions during the discussion questions. So, for now, we're going to begin the Q&A session. So, you can either raise your hand or type your question in the chat. And if we do not get to all of the questions today, Mariia will put Miss Aniqa's email in the chat, so you can always feel free to reach out to her. And there are also some other related papers that Mariia will send the links to in the chat. I don't know if Miss Aniqa, you would like to discuss a little bit more about those related papers.
Those related papers are basically the different kind of theory of addiction, because here they only focus on GABA. So, but there are dopaminergic theory, acetylcholine theory, like different kind of excitatory inhibitory neurotransmitters. So we can't only say by one theory that this can be uh, it's proved that paternal addiction is related to this. So we have to check different neurotransmitter, as I think Ella mentioned. So I agree with that we should check different kind of neurotransmitters. Also, those papers are basically about that. If anyone is interested, they can just go through it.
So feel free to explore those related papers if you're interested. And if you have any questions right now, feel free to come off on mute or type them in the chat.
Yeah, I think one person wrote that I think scholars, Mariami, that focus on animals that are anatomically close to human, like monkeys. Yeah, I agree on that, but uh, I think it's very difficult to do research on monkeys also because then you have very small cohort numbers because it's you will not have much data from monkeys, but of course it's more relevant. I agree on that.
I think Ilya might have a question as well.
Yeah, about the rest of the research. Well, this one was about the drug abuse and how drugs do affect. But are there any other specific related research topics, so such as like this one that we're discussing here at Zoom? Will Simply Neuroscience be able to put? Like this one was about drug abuse at both neurological and genetic factors that we all were working on and were researching on. But what exactly about some things like neurodegenerative diseases, neurodegenerative bios, because those have some hot topics that all of communities, both academia and industry are working on. So, like things that also affect neurotransmitters, things that affect the anatomy, like ALS or any other sophisticated stuff. Are there going to be any events like that going to be in Zoom Live as well?
That's a really great question. Miss Anika, would you like to answer that?
I think he was asking about the other neuro. I think it will be interesting to. I think there is paper about like Alzheimer and Parkinson disease, which can have effects on your next generation, like genetic influence. I think there are already research paper about this because I think in Alzheimer there is one of the because it's the reason is not definite or certain, but one of the main reason is genetics. So I
Yeah, I
think there
know,
are,
because there are some
yeah,
other genetic factors, like for example, Alzheimer's. You mentioned it's
yeah,
APOE4 because it's
yeah,
APOE4 that is causing the aminoplex to go to an
mm-hmm,
underlying
yeah.
mutation. But and there are certain factors about it. So I thought maybe discussions like that, just like how exactly we read the research right now on Nature about the drug abuse that we came here for discussion, maybe that could be also something much more challenging too, because just like drugs, that is a much more and that's much more fun and cooler based on industry if we're talking about.
Yeah.
I think someone had a question in the chat if we had like a social thing like Discord. So unfortunately, we do not have like a specific social platform just for the Synapse session, since we're still relatively new. But if you'd like to get updates or reminders about future Synapse sessions, you can feel free to subscribe to the Simply Neuroscience newsletter. So the link is in the chat, and we usually send out the newsletter every month and it'll usually be at the top. So you'll have like a registration link. And currently, we are working with the technology team at Simply Neuroscience to possibly have a specific page on the website just for the Synapse session. So more updates about that will be sent soon. Does anyone else have any questions? Or maybe there's like any insights or comments they would like to make?
Yeah, I think I have one question about for me. What research have you done on autism and what findings have you found that connect to autism? The my research about autism is more about maternal diet, like maternal high-fat Western diet. If maternal diet can affect on autism or ADHD like behavior in offspring, and we have some data about this. I mean, I didn't publish yet, so that's why I didn't show. I couldn't show because we are trying to find the connection which if there is any gene responsible or protein responsible for if your mother is on high fat diet during pregnancy and lactation.
I think that's really interesting that you're looking at diet specifically because of the gut-brain connection, and I'd love to
Yeah,
learn more about this topic for sure.
yeah. It's very interesting. I mean, it's very difficult. Of course, I also work on rats, so of course it's not, I mean, you can't just prove with one experiment on one behavioral study. So I mean, I just finished my first year. I had to do a lot. We have till now some behavioral studies, like if we are watching if mothers' diet change their social behavior, if they're more hyperactive or antisocial, something like that. So of course, in uh, animal is different, but this is the starting point from research.
In autism that you're working now, since mostly it's a neurodevelopmental disease that occurs in children, which genes are you right now screening? Because I know that diets have a very crucial point and very crucial effect on genes. The way we eat, the way we have our diet, it
Yeah.
affects our epigenetics. But from research that has done, we know it's still a mystery. Things like not just autism, it's just also not that. It's epilepsy as well. It's one of those childhood disorders that also has different types of it in different brain regions, and each regions with each gene has a specific mutation. But in autism, for example, there are very little genes that we know about, like ADNP, for example, like ADNP. Are you focusing on ADNP, or which diets when you guys study in the genes? Which genes do you guys screen? ADNP, or there was also
There
another
are
gene.
we already have a gene map, so
Mhm.
we just selected because it's not proven. So we still don't have a baseline, but we just selected from previous some paper like Shank or NRXN, those.
Uh-huh.
So
What about
yeah.
epilepsy as well? I know it's still not based what exactly you're researching at, but what about
Yeah.
epilepsy? Did you have formed anything in that?
No, I'm not focusing on epilepsy, but I mean, no, my main focus in this autism and ADHD and this addiction is my like a side project. It's not my
Uh-huh.
PhD project. So
I see.
my
Yeah.
department is mainly drug addiction, pharmacology. So we are mainly try to connect the food addiction, drug addiction, if changes in your pharmacology or your brain.
Mhm. Which region are you now? Your undergrad, you mean, or like what else?
No, I'm in my PhD,
PhDs in neurobiology or neuroscience complete?
neuroscience.
Yeah, neuroscience. Where
Mhm.
exactly?
It's in Poland. It's Polish Academy of Science.
Polish Academy of Science. Yeah, I know that
Mhm.
region. Yeah, thank you so much.
Thank you.
I guess another question in the chat is more personal. How did you start conducting research, and what would you say is the best way to do so?
I mean, I for me it was a long journey because I finished my medical school, so for me it was difficult to switch to research because clinical studies are not research. I mean, you just it's external practice and all. So I had to do a master's again, even though I have this MD because research is something different. So I think it depends on your master project, which area you want to focus. So I was always interested into brain. For me, it's very interesting, and I started my master's, and then I get into this project, which was about stress, how and related to drugs, vortioxetine. So then I just find my way. Then okay, I want to go further. I want to stick to research, and I want to focus on brain. So that's why I end up in PhD.
That's very interesting. Do we have any more questions, maybe in the chat, or if someone wants to raise their hand?
Thing, no. Thing. I guess if we don't have any other questions or comments, we can move on to the final remarks. But just a friendly reminder that if you would like to reach out to Miss Aniqa, her email is in the chat. Okay, so here are some final remarks. I'll be passing the mic to Miss Aniqa.
Thank you. So from this paper or in general, my comments will be this addiction therapy is multimodal. We need to combine pharmacological, psychotherapeutic approach to target the neurobiological and behavioral factors. Till now, 50% of the patient relapse within the first year of treatment, and current drug manage only adaptation and reducing the withdrawal and craving, but it doesn't reverse the neuroplastic changes. And for future, we can focus on personalized genetic treatments and also we can use AI for behavioral studies, neuromodulation, and by that, I think we are hopeful that we can find a novel drugs that can transform to addiction therapy.
And these are just a couple of other friendly reminders. So the recordings of Synapse sessions will be uploaded to Simply Neuroscience's YouTube channel. So all of these relevant links will be sent out by Mariia in this chat. And if you're interested in attending future Synapse sessions, please subscribe to the newsletter to hear about future announcements. If you're interested in becoming a guest speaker at future Synapse sessions, there is the interest form in the chat. And finally, if you're interested in receiving a certificate of participation, we do have a separate Google form that you can fill out. You will have to attend at least 3 Synapse sessions and then submit some brief reflections. So there is the Google form. But yes, we truly wanted to thank you so much for taking the time to come today. And if you have any questions at all, please feel free to reach out to Mariia, me, or Miss Aniqa. Thank you.
I got some question. Just one thing about this certificate. Are you gonna put the link in the chat, or is it gonna be a reply to our email addresses that we have just got?
I'm sorry, do you mind repeating your question?
Yeah, sure. Is my voice okay now?
Yes, I can hear you.
Yeah. So about the certificate, are we going to get to receive an email, or what? Is there any other way to receive it somehow?
So for the certificate of participation, once you fill out the Google form, the awareness team will review your submission and then we'll send out the official certificate via email.
Ah, okay, I understand. All right, I'll try to do that. Thank you so much.
Thank you. Yeah, of course.
Excuse me.
Yes. Hello.
Hi. So I just want to ask, Anika, Miss Anika. I just want to ask, you know, to do many researches. You need to have a critical thinking. So how can she just reach to that degree of the critical
Critical thinking.
thinking? Yes, that make her have the ability to make many researches.
I think for that you have to read a lot of articles and what's going around the world, what is the hot topic, what is more important, and what other people are doing. It's best way to learn is to connect with other researchers, listen to them, read their work, and like join this kind of synapse session. There are like plenty of things now in online. I think now it's easy to learn, like 10 years, 20 years back. So by that you can under your brain will also adapt, what to do for future. You can also do something.
Okay, thank you so much.
Thank you.
So thank you everyone for coming. If you have any questions, feel free to stay and